Hello, everybody. Welcome to actually our last Precisionary webinar of this year. So after this, everybody gets to enjoy the holidays and hopefully have a good break. So today I am honored to host Dr. Sui-Seng Tee, who is an assistant professor at the University of Maryland School of Medicine. And he's going to be talking to us about imaging cellular metabolism, specifically looking at liver. So Dr. T received his undergraduate and graduate degree from the University of Cambridge. He then transitioned to Stanford University and also Memorial Sloan Kettering Cancer Center for his postdoc training. And then he took a faculty position at the University of Maryland, where he currently resides. So his lab currently investigates the biochemical processes governing how different tissues utilize extracellular nutrients. And to do this, a combination of different approaches is used to measure metabolism, from destructive mass spectrometry to non-invasive MRI scans. He's going to tell us a lot about his work today. So current areas in Dr. T's lab, the focus include the role of fructose metabolism in liver inflammation and the ability to detect these changes using minimally invasive strategies. And as you can imagine, that can have a lot of implications for patient care. So with that, Dr. T, I want to hand it over to you. And again, thank you.
Thanks so much, Abby. That's such a sort of comprehensive introduction of what I do. So I won't really share much about the liver and the brain today, but I'll definitely tell you something about imaging cellular metabolism and how we think strategies that can better detect metabolism can help us, learn a thing or two about the use of nutrients in health and disease. So this is the structure of the seminar today. I'll give you a quick introduction on how signaling, growth factor signaling, and the availability of nutrients affect each other in health and disease, and how a lot of the focus today will be on some imaging techniques, specifically in the realms of MR, magnetic resonance, and this sort of new-ish technique. It's not super new, it's been around now, called hyperpolarized MRI, and how we use these techniques to look at metabolism. I will illustrate the paradigm of the connections between growth factor signaling and imaging using two case studies. The first case study, we'll look at imaging of prostate cancer. And the second, we'll look at fructose. So, these two are interconnected by the fact that they're both downstream, they're both sugars, right? Pyruvate's downstream of glucose, and fructose is very similar to glucose, but utilized differently. So I'll keep coming back to this structure to sort of guide us along as we progress through how we sort of look at imaging, how we look at metabolism using imaging. So this paper really shaped how I view metabolism, right? So Chi Van Dang, while he was, I think he's back at UPenn now, but Chi Van Dang sort of summarized this concept really well. If you are a unicellular organism, all you need is nutrient availability to divide and grow, right? So there's a one-to-one output and input between nutrient availability and building biomass in terms of dividing. However, if you are a little more complex, for example, if you are a metazoan with multiple tissues and organs and have many cells, then you need more than a single input to signal growth and proliferation, right? So now you've got two inputs, right? You need growth factors as well as the availability of nutrients to grow and divide, right? So this is summarized here, right? There is in a unicellular organism, diffusion is usually sufficient to disseminate nutrients all around the organism. But if you are multicellular and you're a metazoan, then you need some level of control to sort of divide the nutrients to different tissues and organs. And of course, different tissues and organs have different nutrient demands. And through different states of physiology and pathology, these demands change as well. So this sort of connection between signaling and nutrient availability has guided my view in research, right? And on the other side of the mirror is when signaling goes wrong or when there is overnutrition or undernutrition, these uh, states cause a disruption of physiology, and that's how we sort of move into the realm of disease, right? So to be able to answer these questions about the interconnections between signaling, nutrients, and health and disease, we have tried to use imaging techniques, right? So today we will talk um, in some detail about magnetic resonance. And the way we use this imaging tools is, of course, to look at, to diagnose disease, right? Can we detect disease early, hopefully using ways that are minimally invasive? And of course, to look at treatment response, right? Now that we can detect disease, can we look at ways that this disease can be ameliorated, and can we test the efficacy of the drugs that we have? And finally, of course, in terms of resistance development, right? I think a lot of the oncology world has been moving towards targeted therapies. And the downside of these therapies, of course, is the development of resistance towards whatever mechanism that we target. So I will start by giving an overview about metabolic imaging. I think a lot of us who are in the cancer world will be familiar with fluorodeoxyglucose, PET imaging, right? Positron emission tomography. So FDG is an analog of glucose, right? With a hydroxyl group replaced by a radioactive fluorine. So it acts sort of like glucose, right? Cells will take it in. And of course, cancer cells love glucose, and cancer cells take it in but by large amounts. And now FDG PET is sort of standard of care in a lot of oncologic descriptions, right? So, this is an example of a melanoma, right? A melanoma patient with, multiple mets across the body that has been treated with a targeted therapy, and, it's almost cleared this patient of disease, right? So we can see metabolic imaging looking at how glucose, is utilized by tumors is of great utility, right? So metabolism is very dynamic, and that sort of helps the utilization of metabolic imaging as a tool, right? So what other techniques do we have in terms of imaging to look at processes of metabolism, right? Because remember, a lot of metabolomics today in the research realm is destructive, right? So a lot of us use mass spec as a way to look at how metabolites turn into one another. But of course, these techniques are sort of a one and done, right? You take your sample out, you have to ionize it, and you don't have it anymore, right? The more techniques that we have to detect metabolism non-invasively, I think the more perhaps we'll learn about metabolism. So the technique that sort of I've been trained in is called hyperpolarized magnetic resonance, right? So I think those of us who do chemistry, we know NMR, nuclear magnetic resonance, which consists of a huge magnet and a small tube. And those of us who do chemistry, we put our sample into the tube and we drop it into the NMR machine, right? And it gives a spectrum. And this spectrum comes from the fact that hydrogen or nuclei that are NMR active, there is a spin difference, right, when they interact in a magnetic environment, right? So when there is a very strong magnet, these nuclei exists in two spin states. It is the difference in the spin states that gives us the NMR signal. At thermal equilibrium, at room temperature, at room temperature, the difference in the spin states are very small. So NMR is inherently a pretty insensitive technique, right? But the sort of main innovation in hyperpolarized technology is the fact that we can force these population differences, right? So now the signal difference is amplified because we have artificially introduced a spin state that is um, that has a bigger difference, right? And the way we do this is through a hyperpolarization process through dynamic nuclear polarization, right? So, these are big words, but all you need to know is number one, it is a very cold process, right? So there needs to be cooling, usually with liquid helium. And there is also an electron transfer process, right? Where, electrons, the polarization electrons are transferred to the carbon-13. So carbon-13 is NMR visible, right? And when it's very cold and there is electron transfer, we are now able to artificially push the spin states to a hyperpolarized state, right? And what can we do with this, right? So about it's now about 20 years that this technology has been around. When I was a PhD student in England, the machine sort of looked like this, some version of R2D2. But it slowly progressed to the point where GE, now the company, makes clinical polarizers, right? So there are now about a dozen clinical sites around the world that use hyperpolarized MR injections into patients, right? We do it here as well in Maryland with my colleague next door, Dr. Dirk Mayer, who's more of an engineer and who sort of really understands the physics behind this. I'm more of the application guy. I'm the biochemist, right? So, what are some examples of the things that we can see, and why is hyperpolarized MR so unique, right? So I think a really important distinction here is that, hyperpolarized MR allows you to measure metabolic flux, right? So a lot of metabolic techniques measure metabolic steady states, right? And I think there is a difference between flux, the conversion of one metabolite to another, versus concentration measurements of measuring what's already there. Of course, hyperpolarized MR is also not radioactive, right? The current sort of gold standard is FDG PET, and fluorine 18 is radioactive, right? In contrast, carbon 13 isn't, right? So it's non-radioactive. And of course, finally, there is the potential for multiplexing, right? Because NMR shifts of different molecules appear depending on the chemical structure of the molecule. These signals are non-overlapping, right? Again, in contrast to FDG PET, where all you're measuring is radiation. So there, if you put in, 18 FDG plus 18 glutamine, there is no way to differentiate the both, right? So, the first example I'll give you of some of the applications of this technique is in the realm of prostate cancer, right? So, prostate cancer has traditionally been very difficult to detect using FDG PET simply because of its proximity to the bladder. So, a lot of these injected radioactive compounds are excreted in the urine. So, clinicians are not really able to visualize prostate lesions sensitively using FDG PET. So the molecule that is most commonly used in hyperpolarized MR today is pyruvate. So remember, pyruvate is a 3-carbon molecule that is downstream of glucose, right? And one of the main fates of pyruvate is its conversion to lactate, right? And we all know that the Warburg effect has been described 100 years ago now, right? That a lot of tumors produce lactate from glucose, and a lot of this comes from the conversion of pyruvate to lactate. So the first clinical trials, the first in-patient description of hyperpolarized MR was published about 10 years ago now, right? As you can see, this is a regular anatomical MR of 2 patients with prostate cancer. I'm not a radiologist, I'm a biochemist, but I think the radiologists tell me that even trained radiologists have a hard time telling apart where the lesion is versus where the normal prostate is, right? So this is where people got really excited about hyperpolarized MR as a technique, right? This is an overlay of the anatomical image with hyperpolarized lactate being produced in these regions that are artificially lit up, right? So we're measuring lactate production from pyruvate. So as you can see, this gives us a way to actually visualize the ability of the tumors to convert pyruvate to lactate, and it seems to correlate with where the tumor is versus normal prostate, right? So as you can see here, MR is a 3D technique, so you get, the ability to reconstruct in 3D as well. And you can see the blue here is the control normal prostate, right? The pyruvate gets injected in, it peaks around here, and then it sort of decays, right? While the red is the lactate forming in these regions. The yellow is where the cancer is. As you can see, there is a lot more lactate being produced in the region of tumor compared to normal prostate. We also have, well, it's not us, the folks at UCSF also put a voxel here in the blood vessel where we predict very little metabolism, but we predict a lot of pyruvate flowing in, right? Because the pyruvate is injected IV. So since this, groundbreaking seminal demonstration of the ability to detect lactate production in prostate tumors, there's been a bunch of papers following this up now, right? So again, the folks at UCSF have been able to detect cancer mets, right? So it's not just the primary tumor that you see, you can also see the tumor travel to other places, which again, you can imagine would be pretty helpful to facilitate detection of metastases, right? And also in terms of treatment, right? So, hyperpolarized pyruvate has been used to detect androgen therapy in prostate cancer. So when I was a postdoc at Sloan Kettering, we wanted to ask the question, how were some of these signals present, right? What is so special about the tumor that allows you to see so much lactate, right? And what are some of the biological characterizations of these tumors? So again, we delivered pyruvate. So this is the sort of scheme that we have in all clinical sites, right? The hyperpolarization process takes about 2 hours. So it is pretty slow, right? And now once this is over, a solution of pyruvate is dispensed. And within 40 seconds, we quickly check for pH, for concentrations, and to make sure that it is injectable into the patient. The injection itself into the patient takes about 10 seconds, and then the MR acquisition is about 60 seconds, right? So as you can see, this needs to be real fast, right? Simply because of, this is MR. We're going against physics here, right? That there is a decay in the hyperpolarized signal that is a function of time. So everything needs to happen pretty quickly. So this is the some of the characteristics of the pyruvate solution when it comes out, right? So it's at physiological pH, right? It's at physiological temperature, and we can inject this into patients. So this is an example of one of the patients two patients that we injected pyruvate in, right? So the way this clinical trial was designed was that all of these patients were scheduled to have radical prostatectomies at the end of the imaging. So the whole prostate was taken out. So we were then able to correlate the imaging, right, the MR, with histology. So that gives us a real strong idea about what are the characteristics of these tumors. So, the Gleason score is how prostate tumors are characterized. The higher the Gleason score, perhaps the more aggressive the tumor is. So tumors with low Gleason scores, we don't have much lactate being produced, whereas tumors with Gleason scores 4 or 5, you see a lot more lactate. Right. So here's a demonstration of how perhaps lactate is a biomarker of tumor aggressiveness as well. So we were very excited to see that. Right. It seems to sort of trend by Gleason score. Right. Now this is just a repeatability where some patients we injected 2 boluses to ensure the sort of consistency of the technique. So, I came in where I wanted to ask what mediates this lactate production, right? What are some of the characteristics of the tumor? And I focused on monocarboxylate transporters, MCT transporters, as its name suggests, transports monocarboxylate, which pyruvate and lactate is one of them, right? So um, usually we think MCT1 mediates the transport of pyruvate into the cell, where MCT4 then shuttles lactate out, right? So this is what we wanted to look at, the expression of MCT1 as a mediator of pyruvate transport into cells. And again, these tumors were the whole prostate was taken out. So we were able to do RNA-seq on these tumors. And as you can see, the expression of MCT1 seems to trend with, positively trend with increasing Gleason score. Right, so this is some of the examples of IHC that we then performed. So it's very clear that benign prostatic tissue has pretty low expression of MCT1, but in the regions of the tumor, MCT1 is upregulated, right? So this is an example of a few different patients. So, here is a better sort of spatial analysis of different regions of the tumor, right? So, again, the benign prostate has very low lactate production, right? And also has very low MCT1 expression. So that is why we think perhaps benign prostate tissue doesn't really convert pyruvate to lactate because the pyruvate is not getting in. So, in contrast, regions with higher Gleason score, we also see higher MCT1 expression and higher lactate production, again, perhaps suggesting that in regions where the prostate tumor is growing, MCT1 is upregulated. These tumors take in lots of pyruvate and convert a lot of it to lactate, right? So we have a sort of ability to look at tumors eating pyruvate, if you may. So, I then went on to ask, how is MCT1 controlled, right? So again, remember, as metazoans, growth factor signaling plays a big part in controlling metabolism. So I zoomed in on the PI3 kinase AKT-mTOR pathway. So this is a sort of classical receptor tyrosine kinase signaling pathway where a receptor is activated when there is an extracellular signal. Although in tumors, in many cases, the receptors themselves or the downstream signaling components are mutated. So this pathway is always switched on, right? So when this pathway is switched on, a cascade of phosphorylation happens, right? Which finally leads to growth and proliferation, right? So of course, every sort of go signal, there is a stop signal as well. And the stop signal for the PI3 kinase pathway is P10. So P10 puts on the brakes, right, to make this signaling cascade stop, right? So, in terms of prostate cancer, do we actually see mutations in this pathway? And the answer is yes, right? So PIK3CA, the first component of the signaling pathway, is mutated in 7% of cancers. And a lot of prostate tumors see a deep deletion of P10, right? So this is where the off signal is gone, right? So this is where the PI3 kinase pathway goes wild in prostate cancer, right? So in our cohort of patients, we unfortunately did not get a lot of P10 mutated tumors, right? There were only 1, 2, 3, 4, 5 regions where P10 was lost, right? But even in this sort of really small cohort, you can see this trend where loss of P10 endows a high lactate phenotype to these tumors, right? So, this is where we sort of started to get the idea that the genetics of the tumors may play a role in the metabolic phenotypes of these tumors, right? So, it's led me to this paradigm where there are certain mutations, right? Different tumors, different diseases have different gene signatures, which endow a certain metabolic change to the tissue or organ that we're looking at. And some of this metabolism can be seen using different imaging techniques, right? So I've given you examples of hypopolarized MR here, but I know there are also different members of the audience that use different techniques, right, including microscopy techniques to be able to sort of detect these changes in signaling pathways, right? So, this is where I sort of left my postdoc to take up an independent position here in Baltimore. And, I've since sort of focused on, the three different directions that I started this talk with, right? Can we use perhaps imaging techniques to stratify patients, right? So right now, again, targeted therapy is a very exciting field of research, right? For example, in the PI3 AKT mTOR pathway, there are multiple inhibitors of the upstream PI3 kinase, the downstream AKT, and as well as the sort of effector kinases, right? So when we have such a large, medley of drugs to choose from, how can we select the best patients that might perhaps respond the best to these drugs? Right. So I put it to you that perhaps, metabolic imaging can be a strategy to help select these patients, right? And, this is an example of PDXs, right? So tumors were taken from different patients and were put into mice, right? This is in collaboration with Eva Corey at the University of Washington. As we can see, when the tumors are PTEN wild type, right, MCT expression seems to be low, right? However, when the tumors are PTEN deleted, MCT expression seems to be high. So this gives us some support for, the use of hyperpolarized MR maybe, where we can hypothesize that tumors that produce large amounts of lactate from pyruvate will probably respond better to PI3 kinase inhibitors, right? So, besides stratification, I've also done some work in treatment response, right, where we treated a bunch of PTEN null cells with MK2206, which is an AKT inhibitor currently in clinical trials, right? We can see that treatment of these cells with this inhibitor very effectively suppresses AKT signaling, right? So this is an example of on-target inhibition. And when I put these tumoroids, right, so the aggregations of tumor cells, spheroids, in alginate and encapsulate them so that we can put them into an MR machine, we see much less lactate produced when we treat them with MK2206, right? So this is another example where perhaps we can use hyperpolarized MR to look at treatment response, right, where PI3 kinase pathway targeted therapies result in a suppression of pyruvate conversion into lactate. Yeah. So, again, this is where perhaps some, organotypic cultures, such as, the use of precisionary compresstomes to sort of slice tumors can be used to sort of prove this concept further. Right. So I've also done some of this in MR compatible bioreactors, right? So I put, so these are individual spheroids of the cell cultures and encapsulated in a 3D matrix, right? And we can sort of stain them because, of course, the hyperpolarized MR is completely non-destructive. You inject pyruvate in, the cells are still completely fine. Yeah. So I've been working with some engineers to make a compatible bioreactor so we can fit this into the MR machine and hopefully we can see metabolism there as well. Right. So finally, we've also done some work in resistance where we hypothesize that, when cells become resistant to PI3 kinase inhibitors, the metabolism converts back, to when it was not treated, right? So basically, resistant cells will have high lactate, right? So we're sort of in the process of doing this, where I've worked with Rena Peters, who is the head of our translational core, where we are now sort of switching to breast cancer, where we make cells resistant to different either PI3 kinase inhibitors or different inhibitors, right? And I have a student here who's an intern, and when we do Western blots on these resistant cells, right, we see MCT1 is completely sort of reduced instead. Right, so we are really not sure what is going on now, but we definitely see a metabolic phenotype when cells get resistant to PI3 kinase inhibitors, right? So this sort of ends my first part, the first case study, right, where we hypothesize that targeted therapies induce a metabolic change, right, and it's the same with drug resistance, and metabolic imaging can be used to sort of look at these phenotypes non-invasively. Yep. So again, as I've said, future directions include tumor slices, right? I started some of this while I was in Sloan Kettering. We're trying to translate them here as well, where we grow these tumor slices in these inserts in 6-well plates, right? It rotates to ensure sufficient oxygenation. And we can take these slices out, we can stain for DAPI, and this is calcine AM, a live cell stain, and we can see that these tumor slices remain viable. Right, so let me move on to the second part, right? So in the first part, we talked about pyruvate conversion into lactate, right, as a sort of downstream effector of the glycolytic pathway. But now we're shifting gears to fructose instead. So fructose is very similar to glucose, right? The only difference is, now we've got a keto group right here in fructose. So fructose is a keto sugar. But the pathways that fructose is metabolized into the cell is pretty analogous to glucose, but with a few distinct differences, right? So the transporters that take in fructose is different from glucose, right? And the downstream enzymes as well, right? So while both sugars are phosphorylated when it enters the cell, the enzymes that phosphorylate fructose and glucose is different. Right, so fructose can be seen as a specialized metabolic pathway that is only limited to certain cells and tissues, right? So while glucose is broken down in most cells and tissues, fructose metabolism is limited to the liver, kidney, and small intestine, right? So, this specialized metabolic function is, again, a adaptation to the fact that different tissues and organs have different metabolic demands, right? So I really wanted, to go in and ask what mediates these differences in metabolism. So the way to do this was I focused on KHK, right? The first enzyme that phosphorylates fructose into fructose 1-phosphate. And I asked the question, where is KHK expressed through development and through different organs, right? So this is a whole mount slice of an embryonic mouse, of a fetal mouse. And as you can see, there is very little KHK. So it seems that ability to break down fructose is switched on sometimes after birth, right? So, and a lot of this is localized to the adult kidney, the adult intestines, and of course the liver, right? So the conclusion here is that KHK is temporally and spatially controlled. And it's the same in the human, right? Where the liver, intestines, and kidneys express high levels of KHK that is not seen in a lot of other tissues. So I wanted to ask, well, if the liver is an avid site of fructose metabolism, what happens in liver cancer, right? So I found out that in many liver cancer cell lines, KHK expression is very low compared to the normal liver, right? So C3A and HepG2 are hepatoma cell lines, so they are less aggressive, and they still retain some ability to break down fructose. But in HCC, sort of more aggressive hepatocellular carcinoma, the ability to break down fructose is almost completely gone, right? It's the same as well in the intestines, where intestinal cancer cell lines express much lower KHK compared to normal intestines, right? So we also measured this functionally, where we used a fluorescent analog of fructose, right? And as we can see in HepG2 cells, which is the hepatoma cell line, there is still some ability to take up fructose, which is lost in HCC. So, I was at Sloan Kettering at the time, so we were very lucky to have, Scott Lowe, who's an expert in generating liver cancer models. And regardless of of the genetic background of the cancer, it seems that, most of these cancers exclude KHK expression, right? So again, KHK is lost in HCC. And not only is KHK lost, the ability to break down fructose is also much lower in liver tumors, right? Again, here we use mass spec, right? We can see that, there's much less fructose in the livers of, of these mice with tumors, right? And again, F1P, fructose 1 phosphate, which is downstream of KHK, is also suppressed. So how about, in liver cancer patients, right? So this is an example of RNA expression in liver cancer patients, right? We can see that in I cluster 1, which is just a clustering of patients based on the genetic profiles, KHK expression is significantly downregulated in I cluster 1, right? So significantly, I cluster 1 patients perform less well, right? They have tumors that are more aggressive, right? So, besides KHK, we also see that the transporter of fructose in the liver, GLUT2, SLC2A2, is also downregulated, as well as aldolase B, right? So, which is the enzyme downstream of KHK, right? So, fructose 1 phosphate is cleaved into 2 3-carbon molecules by aldolase, and this enzyme is also downregulated. So, what I concluded from here is that it's not just KHK that is downregulated, it's the whole fructose metabolism pathway, right? The transporter is down, the kinase is down, the aldolase is also down, right? So then we went in to look at human patient tissue, right? So normal liver expresses high amounts of KHK, but HCC, right, liver cancer expresses really low levels of KHK, right? So now we have a paradigm where we hypothesize that liver cancer loses its ability to break down fructose, right? So we went on to ask, can we use fructose as an imaging biomarker, right? So, in this paradigm, we inject mice with fructose, and we assume that the normal liver will break down fructose into lots of F1P, right? And this should be lost in liver cancer, right? So, using MR, we can pretty clearly distinguish F1P from fructose by, the chemical structure, chemical signature of this molecule. So, unfortunately, as I've mentioned, hyperpolarized MR is limited by physics, right? The hyperpolarized state of the carbon only lasts, at most 1 minute, right? It very highly depends on the molecules, right? So the lifetime here is called the T1. The T1 value determines how long the hyperpolarized state is, and it's affected by many different factors, including how many protons there are, how many hydrogens there are in the molecule. So unfortunately, fructose is full of protons, and these protons sort of shorten the lifetime of the hyperpolarized product, right? So, we had to find a way to replace these protons with deuterium to lengthen the T1 so that we have more time to image, either the mouse or the patients, right? So, I had a very smart chemist who was working with me at the time, and he figured out a way to deuterate, to replace all of the protons with deuteriums to lengthen the lifetimes of the fructose, right? So as we can see here, when we replace the protons with deuterium, we really lengthen the lifetime of the fructose molecule, right? So we then went in with this deuterated molecule, injected it into mice, and as you can see, the normal liver produces good amounts of fructose 1 phosphate, while the liver cancer tumors produce much less, right? So again, this was recently published. And when I came to Baltimore, right, I really wanted to ask the question, well, now liver cancer occurs in the background of non-alcoholic fatty liver disease, right? So in the past, a lot of liver cancer came from viral etiologies, right? Either hepatitis B or C, but now NASH, liver inflammation, is now the main cause of it's a leading cause. It's not a main cause yet, but it has seen the largest increase in the incidences of liver cancer, right? So a lot of experts predict that in the future, NASH will be a leading contributor of liver cancer, especially in developed countries, right? So here we predict that when the normal liver is exposed to diets high in fat and high in fructose, and it progresses to inflammation, NASH, non-alcoholic steatohepatitis, KHK expression is downregulated, right? So here was the main hypothesis that I was working with. When the liver gets injured, it shuts down fructose metabolism, right? So to answer this question, I collaborated with an expert in NASH, and Dr. Yu here uses a genetic model, right? So this model is free from diet-induced effects. It's free from metabolic effects, right? It's a single gene is knocked out in this model. This gene is called CGI58, and it's involved in lipolysis. And once this gene is knocked out, the liver now stores large amounts of fat, which lead on to inflammation, right? So again, this is um, uh, a IHC with KHK, right? So when we try to detect the enzyme that breaks down fructose, we see that feeding high-fat diets downregulate KHK, so this supports our hypothesis. And in the CGI58 knockout mice, the phenotype is exacerbated, right? So when you lose the enzyme that breaks down fat, your KHK is down, right? So when the liver is injured, it stops breaking down fructose. So, besides MR, I also wanted to ask, can circulating fructose in the serum be used as the biomarker for, injured liver tissue? All right, so because right now it is really hard to predict how to predict or to diagnose patients that have simple steatosis, where the liver is just fatty, versus patients who have inflammatory liver conditions such as NASH, right? So here is perhaps a minimally invasive way where how much fructose is left in the blood can tell us something about the state of the liver, right? So again, we think we can inject a healthy mouse with a bolus of fructose, and let's measure what's left in the serum, at several time points after, right? So the hypothesis is that if the liver is injured, you will see much more fructose floating around because the liver is metabolizing much less, right? So this is exactly what we see, right? So when we use mass spec to measure fructose in the blood, so we use a carbon-13 label fructose again, so we can see that there's much more circulating fructose left in NASH mice. So when the liver is injured, there is more fructose floating around because the liver breaks down much less fructose, right? So we can see fructose being converted into lactate as well. So in NASH liver, there is less conversion of fructose into lactate, pyruvate, and alanine, right? So the injured liver metabolizes less fructose. So again, we're coming back down to where I started, right? My interest has always been to look at how signaling controls metabolism, right? And in terms of fructose, we actually don't know a lot about this, right? So the classical paradigm of glucose and growth factor control is very well elucidated, right? So we know that insulin binds to a receptor on the cell, right? So insulin is a growth factor. And when it binds to a receptor, it signals through PI3 kinase AKT to promote the cell to take in glucose, right? But to our surprise, in terms of fructose, we really don't know what are the growth factors that control fructose metabolism, right? So I went on to ask the question, well, if fructose is controlled by an extracellular growth factor, it has to respond to extracellular challenges, right? So my postdoc here showed that KHK expression is definitely influenced by its extracellular environment, right? Where he challenged cells with FBS, right? So FBS is a soup that contains many growth factors. And in the presence of fructose, KHK is slowly upregulated, right? So, here is evidence that KHK expression, fructose metabolism, is responsive to extracellular factors, right? And we were also very surprised to find that when you treat HepG2 cells, so these are the hepatoma cells that do take up fructose, with media that cancer cells were grown in, right? So conditioned media from cancer cells are able to suppress KHK expression, right? So it seems that cancer cells produce some kind of factor that forces liver cells to downregulate fructose metabolism. So this was a huge surprise to us, right? So there were really no reports of an extracellular secreted factor produced by cancer cells that are able to suppress KHK expression, right? So, after a bit of searching, we found that insulin-like growth factor, IGF-1, appears to be a growth factor that, when treated, downregulates KHK expression, right? So this is something that we're actively investigating right now, where we think IGF binding to its receptor, IGF-1R, signals through PI3 kinase AKT to suppress KHK expression, right? So I think now, we have a handle on how growth factors control fructose metabolism as well, which I personally think is pretty exciting. Right, so if it's true that there are these extracellular factors that are in play, do these observations extend to fatty liver disease? Right, so I was very lucky to work with Yang Xiao and Mitch Lazar at UPenn, where they have single cell RNA sequencing of hepatocytes, right? So as we've said before, injured liver in terms of NASH, so this is a mouse model of NASH, down-regulate KHK over time, right? So again, this is consistent with what we've seen. But what is surprising here is that the same hepatocytes seem to upregulate IGF-1R, right? So again, remember, IGF signaling antagonizes KHK expression, right? So we see evidence for this as well. So, we're now actively trying to sort of prove these models in more sophisticated, prove this signaling pathway in more sophisticated models. And again, we've treated some liver slices with fatty acid to sort of mimic a steatotic environment. And we're on our way to look at KHK expression and IGF signaling. Right. So I want to conclude here by saying, if you've taken back nothing more than this slide here, I would be very happy, right? That in growth and disease, there's an interplay between growth factor signaling and nutrients that dictates how cells and tissues respond to different metabolic needs, right? So in the paradigm of pyruvate, we see that in prostate and breast cancers, the PI3 kinase AKT mTOR pathway is very much mutated, right? It's been a pathway that has been targeted by many pharmaceutical companies, right? And it can give us a flavor of what different types of pathways control glucose metabolism. In the fructose space, we've shown that in NAFLD and HCC, when livers get diseased, fructose metabolism is a pathway that is very much downregulated, right? So here in the pyruvate space, we look at MCT1 expression. We've sort of we're starting to see evidence where PI3 kinase mutations affect MCT1 expression, and we can sort of probe these mutations using hyperpolarized pyruvate. In an analogous manner, we see KHK downregulation in the liver, right, which affects fructose metabolism. Right. So, I think there's a lot of space for future investigations here, right? So we can do this technique in humans. So, there are many places right now that are incorporating imaging techniques in the clinical trials, and I think this is going to be really exciting. And no one has really injected fructose as an imaging agent into humans yet, but hopefully as we get better tools, we will really be able to do that. Right. So, this is a huge, effort from many labs all over, right? So, Sloan Kettering being one, and UPenn has been a really good collaborator while I've been here. And, we've been very lucky to get NIH funding to do some of this research. And, I now just have one postdoc, right? And so Salah has been such a hero in doing so much of this work here. Right, so I'll stop here for questions. This is just a photograph of autumn in Baltimore. It's gorgeous. So if you guys haven't been down here, please come down here and let's have a chat.
That's right, and you won't have to suffer any humidity, which Baltimore is also famous for. But the change in leaves and change in trees is beautiful in this time of year. Well, I really hope you the people who attend today and registered have about time to visit Dr. T's lab. So as from our group of metazoans here, does anybody have any comments or questions for Dr. T's presentation? Thank you so much, by the way. And please feel free to unmute yourselves and show yourselves. If you don't, that's also okay. Please note that you can also ask questions later. It looks like Dr. Shi, you might have a question. Would you?
Oh, yes, I have a question about first, a wonderful talk. Thank you for sharing the work.
Thanks, Lingyan. We're very happy to see you here.
Yes, so happy to see you again. My question is about the spatial information for metabolic dynamics, how important it is, and what kind of specific question you are potentially you can target for figuring out or mapping out the spatial distribution of metabolic dynamics.
Yes, Lingyan, I think you've brought a really important question here, right? So traditional MRI has pretty good spatial resolution, right? I think at its best, MRI can do sub-micron spatial resolution. But hyperpolarized MR has very bad spatial resolution, right? Because so much of the energy is being used to detect metabolism that I think the best resolution that we have is probably at millimeters. So hyperpolarized MR is not a very good way to answer questions about, spatial metabolism, right? So that's why, I was really excited to work with you to see if we can use some, optical techniques to answer more specific questions about metabolism at a more refined spatial resolution, right? And I'll tell you why, because of course, metabolism isn't mediated by a single cell type, right? When we are looking at tumors, right? There are tumor cells, immune cells, fibroblasts. Hyperpolarized MR is not able to answer those questions, right? We see a lot of lactate. We don't know where that lactate is coming from. We assume that the majority of the signal comes from tumor cells, but maybe the immune component really contributes as well. We know that activated T cells produce lots of lactate, right? That's a classical sign of T cell activation. So I think a combination of MR with optical might be the best way to start answering these questions, because of course, you have methods that are able to detect isotopes as well. So I really think there is space for collaboration here, because you do things that we don't do well in the MR world.
Thank you so much, Dr. T, and I'm so excited about our collaborative work in the near future.
Fingers crossed, Dr. Shi, fingers crossed.
Yeah. Yes.
Wonderful. Wonderful. I really wish both of you the best too in your collaboration.
Thanks, Abby.
Well, if there are no more additional questions, then please feel free to reach out to us. I will conclude our meeting. And Dr. T, I just want to let you know I do have a couple of references that I searched for and found relating neurodegenerative disease in liver studies. I will shoot them your way by the end of today. I've got a lot of meetings, unfortunately.
Thanks, Abby.
And thank you also to everybody who attended today. I hope you all enjoy the rest of your week and the holidays. Take care. Bye-bye.
Bye. Thank you. Bye. Take care.